Cell replication and cell cycles, AQA biology. Key terms. Mitosis. Mitosis produces two daughter cells that have the same number of chromosomes present as the parent cells in each other. Meiosis produces four daughter cells with half of the number of the parent cell. Diploid is a homologous chromosome from a parent cell. Cancer, the inability to control the cell cycle, grows aggressively and invades nearby tissues. Tumor, cells that keep dividing to make more cells. Proto-oncogenes, produce proteins that stimulate cell division. The gene also inhibits apoptosis, which is cell suicide. Tumor suppressors, prevent cell division, leading to apoptosis. Mitotic index, ratio of dividing cells to the total number of cells visible, usually higher in rapidly dividing cells. Prokaryotic cells and viruses. Prokaryotic cells replicate in the following process. They replicate through binary fission, and cells replicate genetic material and physically split into two daughter cells. Binary fission occurs whereby DNA replicates once. However, plasmids can multiply many times, so you might get one in the original, in one formed, and six in the other. The cell gets bigger and the DNA loop moves to opposite sides. Cytokinesis occurs, which is the division of the cytoplasm, and then the cell wall divides, and each daughter cell has one copy of the DNA. Viruses are acellular, and this is the structure of a virus. It's acellular, like I said before, and it's a simple nucleic acid surrounded by protein enclosed in a capsid. And acellular means it's not alive. How do viruses work? Well, they can't exactly reproduce by themselves, so they have to go and invade host cells. They attach their host cells using attachment proteins and inject their DNA or RNA into the host. Then they use their own cell's machinery to replicate, and they replicate and replicate and replicate until the cell is full and it physically can't hold anymore, so it bursts, and the virus is able to spread to other host cells in the body. Mitosis. Mitosis steps. You have prophase, metaphase, anaphase, and telophase. Prophase includes the centrioles, which are proteins that are at the top and the bottom of a molecule, move to opposite ends of the cell. Spindle fibers start to develop. Metaphase causes chromosomes to line up along the middle, also known as the equator of the cell, along the spindle fibers. The spindle fibers arrange themselves along the equator of the cell as well. During anaphase, the centromeres start to divide, separating the pair of sister chromatids, and the sister chromatids are pulled to opposite poles of the spindle fibers. And this makes the chromatids appear V-shaped. Telophase causes the chromosomes to unite, and the nuclear envelope reforms and cytokinesis occurs, which I explained is the same thing with the viruses, with the cell membrane, not cell membrane, the cell not cell membrane, what's the thing called? Cytoplasm dividing as well. Don't know why that took me so long. DNA. DNA is linear and it's wrapped up. It's folded and tightly bounded to proteins called histones. And the DNA bounded to histone is called chromatin. Cell cycle. The cell cycle composes of G1, synthesis, G2, mitosis. G1 involves the growing of proteins. Synthesis is when the DNA duplicates. G2 is the growth until mitosis, and the internal organelles replicate. Interphase is everything, all of the cell cycle except for mitosis, and this usually is about 90% of the cell cycle. And this is a little diagram showing what the different stages are. And the mitotic index, which is the number of cells in mitosis divided by the total number of cells. Now, thank you for watching this video. I know it was pretty short and sweet. And the next video, I'll be talking about the transport of substances across cell membranes. Thank you very much. If you like the video, please like and subscribe. Thanks.